All right, guys. So we have 3 more lectures, and we're gonna pretty much start chapter 13 today. So I know your brains are swollen from the lab. So I'll actually probably will finish a little bit earlier than usual, since I have plenty of time to finish this next week. Put you guys out of your misery and let you guys go out.
Wait, we only have 3 lectures for the whole rest of the semester?
No. Like, what? Like, dude, don't stress me out. No, for the rest of the immune system, right? So we have next week, we have today, and the 2 more lectures next week, and then your exam is the Monday, 11, 12, and 13. All right, so last time we started talking about the line of defenses, human defense. We talked about line 1, which was your barriers. First line of defense is your barriers. Brain. That's where my brain is. Skin and mucous membrane, those are the barriers. So these are the lines of defense that basically protect you from not getting anything inside, right? And then we also looked at some other things that we had, like possibilities of having lysozyme in your saliva, in your tear, in your mucus, in any secretions, having stomach acid, normal flora. So these were all your barriers. Then we had the second line of defense, which we started. That was your non-specific internal inflammation. So we looked at the process of inflammation, how inflammation gets started. Then the purpose of inflammation or inflammatory response ultimately is to repair any tissue damage that you have, and finally, if there is any pathogens there, remove it by the process of phagocytosis, or slow their reproductive rate so that that contamination doesn't change into infection. We looked at the process of phagocytosis. We actually looked specifically at macrophages when we did that, because I chose macrophages because they're the ones that ultimately, as we see today, they're going to be the middleman between your second line of defense and third line of defense. And they're the ones that have MHC1s and MHC2, and if they go through phagocytosis, they will take the antigen of what they have engulfed and place it on MHC2 and become an antigen-presenting cell. All right? Then we talked about possibilities of, in addition to cellular attacks that you can have from those white blood cells that we looked at, neutrophils, eosinophils, macrophages, natural killer cells, we also have some chemicals that can start reacting and getting activated, and they can actually damage. The first one that we looked at was the complement groups of proteins, the complement cascade. Which is the last thing we looked at during Monday's lecture. Complement cascade is a group of blood proteins that interact with one another. One activates the other, and ultimately the purpose of cascades getting activated, or what the conclusion of it, would be a MAC, membrane attack complex, that will poke a hole into cellular pathogens and lyse them. In addition to that, some of those proteins are opsonine, like C3b. Some of the proteins, like C3a, C5a, they can actually induce inflammation, keep inflammation going. They can bring more white blood cells like neutrophils to the area. So those were some of the results of complement. There are 2 pathways. The one that we looked at was alternate pathway. Ignore the particles of C3a, C3b, and all of that at the beginning when you're studying it. Just know this. Alternate pathway brings on complement or activates your complement during inflammation. What is important in initiation of that is C3 that splits into A and B, all right? C3B, and I said to be or not to be, B is the answer. So always it's the B's. So out of those, C3, if C3B finds a pathogen and attaches to it, it will set on the alternate pathway. Eventually, it's going to lead into C3 convertase. C3 convertase's job is to now break any C3 that is in that area. More C3s in the environment eventually causes a change of C3 convertase to C5 convertase. C5 convertase then breaks down C5, which eventually leads to MAC. Study it that way. Once you understand that, then go over, okay, C3 convertase is C3BBB. C5 convertase is C3BB, C3BBB3B. Things like that, all right? How did factor B got split? It was with the help of factor D. Even if you forget factor D and all of that, you know most of the important things about complement cascade, all right? Okay, questions for me? All right, so let's get started with today's lecture. So another complement, sorry, I did not sleep last night. I don't know why, so my brain is kind of halfway on. Another protein that is responsible for a possible attack on pathogens during inflammation is the protein interferon, or the cytokine called interferon. Remember, cytokines are proteins that have a signaling function in your immune system. Okay. Interferon is a cytokine that can attack or cause a change in viral infections. So interferon is for viral. Where complements were for cellular pathogens, interferon is only released in response to viral infections. There's actually three kinds: alpha, beta, gamma. The difference, they all function the same. The difference is like where they're released from, all right? Generally, they are released first and foremost from a virally infected cell. So I'm going to use this image. Let's say a cell gets infected by a virus. Before that cell is taken over by the virus, and it doesn't matter what kind of a virus it is, most of our cells, when they realize that they've been attacked or they have been infected by an internal virus, then they start releasing the production, or they start releasing or producing the protein interferon. Then they release that into the environment next to them. Every cell that we have has on its surface a receptor for the molecule interferon, for the cytokine interferon. So the infected cell is releasing interferon. This is basically like setting off the alarm. Interferon is going to go and attach to all of the neighboring cells in the area of inflammation. And once it attaches to the receptors, those receptors change shape and basically cause a signal inside of the cell. Inside of that healthy cell, basically the signal says this: we have a virus around, so possible infection is imminent. That cell, then what it does two things. It's going to create proteins that will break down single-stranded RNAs. So whether all of the mRNAs that it has, everything, it's going to break that down. In addition, it will shut down the protein synthesis. So nothing is getting transcribed or translated. So protein synthesis is shut down. How that helps with viral infection is this. Now that this cell that has the signal, when the virus goes and gets inside, first off, if it's an RNA virus, The enzyme is already there, and hopefully it will damage the viral nucleic acid, that was single-stranded RNA. If not, the virus cannot turn on protein synthesis. So it has to follow the rules of the whole cell. It's kind of like in those sci-fi movies when aliens take over the ship and they put the auto-destruct. Well, they can't stop it because they don't know the code, so they're going to get blown up with the ship. Same kind of a concept. They come in, and the most important thing they need is the process of protein synthesis, because they have to synthesize and assemble their parts. So when that's down, that virus is stuck in there, not being able to replicate. Now, my question to you is this. Can the process of protein synthesis shut down indefinitely in a cell? Why not? What do you think will happen? If you shut it down completely, what happens to that cell? It dies, exactly. So, it cannot do it indefinitely. This is why your cells would slow it down, but they can't do that because otherwise they will die. So they eventually will turn back on the protein synthesis. When your cell is in stress, one of the biggest signals that the tissues or the cells in the tissue, basically when protein synthesis is done and it's not working or the cells are not functioning normally, one of the signals to alert you is pain. This is why some viral infections can cause severe pains in the muscles and joints. Those are the cells that you're using a lot. So you're still walking around, and every time you use that, your muscle is moving. The muscle cell wants to exist and function, but interferon is shutting it down. So its way of telling you, I'm not healthy, is by giving you the pain. So you're not getting the pain from flu, because flu causes pain. It's the interferon, all right? So eventually it gets back. So let me show you the animation for that. And before that, again, what's the difference is interferon gamma is, for example, released by T lymphocytes and natural killer cells. Alpha and beta most often by the infected cells and macrophages in the area. So that's the only difference. Otherwise, they function exactly the same. Hopefully it's not too loud.
When a cell is infected by a virus, the virus enters the cell and produces structures that are not found in uninfected cells. The presence of this viral material signals the cell to produce interferon. The interferon moves out of the cell and attaches to receptors on nearby cells of the same type. The cell that produces the interferon is unable to save itself. The virus replicates in this cell and then moves out to infect nearby cells. The nearby cell that already has interferon bound to its surface responds in several ways, including production of enzymes that degrade messenger RNA and prevent protein synthesis. Thus, a virus can attach and enter the cell, but completion of the viral replication cycle is prevented.
But obviously it can't do this the whole time. You can keep shutting it on and off, but if it keeps it going on, the cell is going to die. All right. Next one that we will talk about is a group of little tiny proteins that are released by neutrophils, natural killer cells, and lymphocytes. These are called defensins. Defensins are these little tiny round proteins. They interact with the proteins on the surface of pathogens. And for example, for a bacteria, they prevent them from going through process of proper transport of material. So they interfere with the proteins. They might interfere with the glycoproteins of a virus. All right. So complements, interferons, and defenses. Last thing we're going to talk about with regards to inflammation is fever. In many cases, if you have a severe inflammation and you have a lot of inflammatory inducing chemicals being released, some of these, like for example, if you have interferon, a lot of interferon, that triggers the fever. Some other cytokines actually can trigger fever. Histamine even. People who have allergies, we call it hay fever, they do feel a little bit warm when they have a severe, even like seasonal allergies. They feel warm, so their body actually increases the temperature. That's called fever. First off, how does the body regulate the temperature of the blood? This is done with the help of hypothalamus. So your hypothalamus is like a thermostat, and it sets a specific body temperature for each person. Some people run around right 37 degrees Celsius, some people run a little bit cooler, some people run a little bit warmer. But how that is controlled by the temperature of the blood. When you get really hot, what happens to your face? You turn red. So hypothalamus opens up all of the arterioles to the surface of the skin and sends the blood to the surface of the skin. With the help of sweating and blood getting, your skin getting basically brushed by the air, it cools the temperature of your blood and it goes back and hopefully your body temperature goes down. When you are cold, it's exactly the opposite. That's why a person turns white, okay? Blood is prevented from going to the surface. It remains inside, and sometimes the body, like those little tiny shivers and things, your body actually, hypothalamus forces some of your small muscles to go through contraction and contract and relax, so they will burn ATP and they can produce the byproduct heat to warm up the blood. Hypothalamus's set can be changed with the help of a group of chemicals called pyrogens. Pyrogens are a group of chemicals that affect the hypothalamus and they cause a change in the setting for it. And in most cases cause a fever. You can have exogenous ones, so these are chemicals associated with the pathogen itself, like a pathogen's flagella, the capsule of it, can actually be identified in blood, and that on its own can trigger hypothalamus. Endogenous one, these are cytokines, interleukins, interferons. These can actually cause hypothalamus to increase it. So hypothalamus changes and says blood temperature goes up. So most of your blood comes inside. By the way, for you guys are used to Fahrenheit, so in most cases, your human temperature should be 98.6, right? Slight body temperature is not even considered a major fever. So like fever of 100 degrees, if you go, many doctors are like, just let it be. Don't even worry about it, just watch it, make sure it doesn't go higher. But higher fevers like 101, 102, 103, 104, those are very dangerous. Chills that you get, chills mean actually your fever is extremely high. The person would be very warm to touch, but they feel cold. Why? Because the blood has been kept away from their skin for so long, their skin feels ice cold to them. But when you're touching them, they are actually very hot. They get the chills. Many people, by mistake, what they do when a person has those severe chills, they actually cover them. That's dangerous. That can increase the temperature even higher. Some of the things that you should do actually is cool them down by using cool towels on their legs, putting their legs in cold water and rinsing the feet with cold water. It sounds cruel, but it actually lowers the temperature. Now, why do we need that? It's because of a couple of things. Some of the cells and some of these proteins that are released by white blood cells actually function way better at a higher temperature. Okay. Second thing, some of these pathogens actually have the 98.6 degrees or 37 degrees as their max limit of high temperature. So when your body temperature keeps going higher, their reproduction rate goes actually lower. So it affects stuff. That's why fever is good. However, too much of a high fever is not good because it can damage our own enzymes. So you see seizures in some people, can lead to death. So always have to keep an eye on it. Bless you. So, inflammation. This is what we have. This is the end of the inflammation. So, I love this cartoon. Heat, defense, swelling, pain, and loss of function. So, that's inflammation. All right. Okay. If inflammation doesn't work, and the pathogens can still increase in number, then now you have an infection. So you probably have more signs and symptoms and all of that happening. And this is why it will take a while and you have the incubation period. This is why your body actually has to go through this before it starts healing itself. In some cases, it won't be able to. But it takes a while. The body waits for inflammation to see whether or not it's working or not. And then also what ends up happening is if it's the first time that you've been infected with something, the third line of defense and the cells that need to get activated, they are very late getting to the party. So that's why it starts. You have signs and symptoms. Now let's take a look and see how does your third line of defense get activated and what does it do? The key thing for activation and communication with the third line of defense is your antigen presenting cell. So that's the macrophage that is now presenting on its MHC2. So this is the one. Macrophages take the bacteria in, or in this case is a microbe, take it in, break it down, place it on their MHC2, and they release cytokines that bring on your third line of defenses cells. So let me show you animations with regards to antigen presenting cells. This animation that I show you, it's going to talk about third line of defense and kind of go through the whole defense. Some parts of it are correct, some parts of it are like, oh, okay, you need more, but, so don't worry, it's not like that's it, I'm not going to go through it. Let's take a look at this, and then I'll tell you guys a bit more.
Activation of the immune response typically begins when a pathogen enters the body. Dendritic cells or macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. These cells with antigen fragments displayed on their surfaces are called antigen-presenting cells, or APCs. An antigen-presenting cell interacts with a T helper cell that can recognize that particular antigen. During the interaction, the APC releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation and activation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths: cell-mediated and antibody-mediated immunity. Cell-mediated immunity involves cytotoxic T cells. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. Normal cells of the body that become infected can display antigen fragments from the pathogen on their cell surfaces. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of these infected cells bind to them and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. Antibody-mediated immunity involves B cells. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of cellular pathogens, marking them for destruction by macrophages. They can also neutralize toxins or viral particles. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
Like I said, there are a few mistakes there, but we're going to ignore it because it did a good job of giving you a summary. Okay. All right. Third line of defense is a specific response, meaning that it will be unique for each pathogen. It is also adaptive. The adaptive means that based on the information it receives, it can decide, do I need just to release antibodies? Why? Because the pathogen is just found outside of my cells. Or do I also need to activate my cytotoxic T cells because I have internal infections too? So that's the adaptive part. And then it can produce memory. All of the cells that are involved here will produce memory cells, and large numbers of them that can get activated faster and therefore bring us immunity. They are very specific against antigens. Okay. Here is where we're going to go on a side tangent, and I don't have a slide for this. Cells that are involved in your third line of defense, they're collectively called lymphocytes. We have three kinds of lymphocytes. We have T helper cells. We have cytotoxic T cells, or T cytotoxic cells. And finally, B cells. These are all the three lymphocytes important in your third line of defense. Helper T cell's job is the coordinator. This is the one that speaks to the macrophage. So it is the coordinator, it is the organizer. This is where APC connection cytotoxic T cell is responsible for what we call cell-mediated response. It kills infected cells, so it's infected cells. It gets rid of those. B cells is humoral response, or we also call it antibody mediated response. This responds to pathogens outside, in humor, in the fluids. That's why we call it humoral. Remember, humor is blood, lymph, interstitial fluid. Those three fluids that you can find in your body, they're called collectively humor. All right. All lymphocytes are made in your bone marrow by a group of specific stem cells. They are all nucleated, and they will be made as soon as you are born and your marrow becomes active. They are made in a groups of 3. So you have a helper T cell that has a cytotoxic brother or sister and a B cell brother or sister. I refer to them as my 3 musketeers, even though 3 musketeers were actually 4, but 3 musketeers. They will attack the same thing. So that T cell, helper T cell, will recognize a specific antigen. So they are antigen associated. So they respond to specific antigens. And remember, antigens of each pathogen is unique in shape to that pathogen. Even in strains of different, like for different strains of COVID, what makes them different, and that's how you can actually get sick again, is because the antigen of one strain can look different than the antigen of the other strain. So it's very specific to it. So when you have a helper T cell, when your bone marrow starts producing them, it will produce a helper T cell that would recognize a specific antigen, and that one, and there's only one of them is going to be made, only one. And that will have a cytotoxic T cell that finds the same antigen, and it's going to have one single helper, one single B cell that responds to the humoral. What makes them different than the rest of them, the rest of the helper T cells and B cells and all of that, is in this group, they will have specific receptors on their surface that recognize antigens. So they will have receptors for antigens on their surface. All of these receptors, first off, there is a large group of genes that you have in your body that are responsible for creating these receptors. The way it works is basically like this. Your bone marrow says, I'm going to make helper T cells. Makes a lot of helper T cells. Then they send it to this, again, crazy image that you can have. The helper T cells are put in a bucket and they're sent to the receptor department. In the receptor department, they have one artist and they've said, all right, look at all of these genes that we have. Each of these genes will produce a protein. Your single job is this. Take one of these helper T cells, choose whatever protein you want from up here, You don't even have to use the whole entire. You can use a piece of this one, a little piece of that one, the entire the other one. Put them together and create an antigen binding site that will recognize one specific shape. And stick that on this helper T cell. Take a B cell, put the same receptor on that one, take a cytotoxic T cell, put receptors on that one. Now you have your three musketeers. Put it away. Take this recipe, put it away. Don't ever make it again. You've made it once. Create another receptor. All right? So all receptors, actually, the way they are set is this way. They will have an area that we refer to as constant region. For most us humans, this region is the same. So it's made up of certain proteins. It's a constant region. Think of it as a lollipop stick, or your cone that you buy for, or you request for your ice cream. All the cones look the same. But what makes your lollipop different than the other one is what sugary stuff is put on there, or what flavor of ice cream you want to put in yours. One of you says vanilla, the other one says chocolate. One says vanilla with chocolate chip, the other one says mint chocolate, so on and so forth. So cone is the same. What's inside at the top is different. So constant region for all receptors are the same. What is different, and that person is responsible for making, is what we call a variable region or an antigen binding site. And they have different shapes. So for example, let's say one of them looks like that. It is capable of recognizing a molecule or an antigen that will fit in there. So this will be the antigen binding site, and it is the variable one. You have another receptor. That one is going to recognize this shape. You have another one, so constant region is the same. You have another one that recognizes this shape. So see how each receptor is unique. That's what makes one T cell different than the other T cells. All T cell receptors look like this. So we refer to them as TCRs, T cell receptor. Cytotoxic T cells and helper T cells, their receptors look like that. For B cells, their constant region is a little bit different. It looks like this. They will have two antigen binding sites. So this is a B cell receptor, and it will have two antigen binding sites. Other than that, the constant region is the same, and that's going to be the same. So let's look at it here. Be covered by that all over. they're going to be covered by that. Your body will make 3 musketeers continuously throughout your life, and will create, because you have a large group of genes and proteins that combined together can make variable antigen binding sites that look totally different. You can actually have millions of these sets, 3 musketeers. And in each of them, what's different is just their antigen binding site. Some of these lymphocytes will never, ever find their antigen. Your body doesn't make this in response to, oh, I have a virus in, and this is what the antigen looks like, so let me make something that matches it. You either have it or you don't have it. But you will make so many different ones that minute differences between them. You will have the sets, and some of them, like I said, will never see their antigen. But if they see their antigen, now they become active and they will replicate. So you have single helper T cell that is naive, meaning it was never activated. You have a single cell-mediated, cytotoxic T cell that is naive. That is a twin to this one. You have a single naive B cell. If you don't get a pathogen that has that antigen for them, these 3 will remain just 3. But the second helper T cell of a specific antigen binding site finds its pathogen and becomes active, that thing, like you saw in the movie, is gonna basically get fully activated, replicate like crazy, and create hundreds and thousands identical clones of itself. Then we have clone wars. Okay? Same thing with cytotoxic T cell, same thing with your B cell in that group. If they need to get activated, they will produce thousands of clones of themselves. And each of them now start like a sniper and keep targeting that specific antigen. Okay. So does that concept make sense? Jason?
So like inside of our bodies, there's only one of each type, like a single?
Yes, unless they were activated. If they were activated, you create thousands, and some of those thousands, and I'm talking hundreds and thousands, some of them will get into the war immediately. Some others are reserved. They're your memory ones.
Is it possible for like a cell, like for the antigen to be in your body and just misses it because there's only one? Or is it able to like find it?
It will find it. That's why it takes a while and you get sick. Sometimes it is very hard to find it, so we don't have maybe something exact. It takes a long time, and that's, in the old days, it could have killed someone. Why? Because it took a long time for that single one to find it. See, the thing is, when your APC is there, and your APC, your macrophage, is sending that signal, all of your helper T cells will line up. When you're younger, all of them are single ones. So eventually one of them gets there. But when you get to this age, you have been exposed to so many pathogens, and some of them have created memory helper T cells. When your APC starts going, that memory helper T cells, they line up too. And there's a ton of them, and they get there faster, they respond. So that single one that needs to get activated is way down the line. So it has to wait for its turn to go to the helper T cell and say, let me see, am I the one? And then it gets activated. But once that one produces memory cells, you bet it will find a macrophage somewhere, and it will get to it. fast, and that's when you're immune to it. So for COVID, basically, this is what we had. COVID was a total novel thing, right? But we were able to fight it. Some people not that well. Some people actually, we saw their immune system fought it too well, and it caused them to have like lung issues, damage their own lungs and hearts and all of that. So they would die from it easier. But we all had it. Obviously, the helper T cell for it was somewhere in our body, and we responded to it.
I was just wondering, since we're born with these already, does this have to do with what our grandparents, great-grandparents were exposed to, or does everyone
No.
have this same set of hundreds of thousands of them?
Actually, no. You make it.
Okay.
You make it. It's based on the genes that you have. That can be part of it. But because your mom has immunity to something, that means you don't have it.
Okay.
Because based on your genetic, hopefully you make the helper T cell.
Right.
But if you didn't make it, you can't fight it. That's what people with immune deficiencies, they cannot make these properly. A person that gets AIDS, HIV attacks the coordinator, the commander-in-chief. So these two guys might even see it, but because the commander-in-chief is dead, they kind of sit there and they go, should we, should we, should we, should we? They can't do it. When a person gets a bone marrow transplant, that person, up to that point, whatever immunity or vaccination they had, because their own bone marrow gets totally destroyed, and they get transplanted by somebody else's bone marrow, and this is where the memory cells and all of that is coming from, then they get that person's immunity. And in some cases, they need to be vaccinated all over again for some of these diseases. So if you had chicken pox and now you have immunity for it, you got the bone marrow from a person who's never seen chicken pox in their life, sorry, you will get chicken pox. So they actually take good reports, hopefully from the person they got their bone marrow. All right, so it sounds like you guys are all getting it. Now tell me this, one of them is a helper T cell, one of them is a cytotoxic T cell. B cell, you can totally, a little bit, the receptor is different, right? So you can distinguish from what it looks for, okay? How can you distinguish these two? Can you at this point in time? With what I've drawn up here, is there a difference? No, and they are actually supposed to be different. Your B cells are supposed to respond to the pathogen. So to the whole pathogen on the outside. If it's a virus, your B cell looks in the fluid and finds the virus and attaches to it. So the antigen is on the pathogen. Finds the bacteria, attaches to that. Okay. Helper T cell, however, has to communicate with your antigen presenting cell. It is the antigen presenting cell that says, hey, come here, this is the antigen you're looking for. Where is the antigen being presented on the macrophage? Do you guys remember?
MHC2.
MHC2, correct, both of you. So MHC2. So helper T cell has to respond to a foreign antigen on MHC2. Cytotoxic T cell kills your infected cells. Your infected cells, virally infected cells, where do they present the viral antigen on the surface? MHC 1 or 2?
1.
1, exactly. They don't have MHC 2. The only one that has MHC 2 is macrophage and B cells. We'll see them. So obviously, we have to come up with a way to differentiate these guys. And the body has done it. Next to every single receptor on a helper T cell, I'm going to use coloring. Every helper T cell, right next to that receptor, they have another receptor, another protein called CD4. So these are CD4s, these we refer to as TCRs, all right? CD4's job is to recognize and confirm presence of MHC2. This is a term I use. So a helper T cell has to do a double handshake with the antigen presenting cell, with the macrophage. Its TCR, the antigen binding site, has to say, I see you, the antigen. I am a match for your antigen. It won't do anything, that's the first handshake, until the second handshake goes and CD4 goes and grabs MHC. Are you MHC1 or MHC2? If you're MHC2 and my TCR matches you, fantastic. Double handshake, we are activated. If this is on an MHC1, both hands are retracted and helper T cell moves back. I am not supposed to respond to you. Okay. Cytotoxic T cell has its own unique second hand. That's called CD8. CD8 responds to MHC1. CD4 responds to MHC2. This one is even more important. So a cytotoxic T cell goes and, let's say it's this one, goes and says, oh, fantastic, my TCR matches this antigen. CD8 goes and says, grabs the MHC and says it's MHC2. It will freak out and totally step away, more so than the helper T cell. Why do you think this is bad if it recognizes MHC2 and says go ahead, attack? What is it killing?
The macrophage.
The macrophage. So cytotoxic T cells CD8's recognition of MHC1 versus MHC2 is even more essential. It has to confirm, double confirm, antigen is on MHC1. This is an infected cell, let's kill it. If it's MHC2, step back, because it's either a macrophage or a B cell. Your very important immune cells. So that's how you distinguish them. B cells you don't worry about, because they find something on the outside and they won't react until they get a confirmation. Good, everyone? I don't have any slide for this because I don't know how to explain this on a slide. It's scattered through all of my slides. So, B cells, so humoral cell, they respond to anything that is in your body fluid. Their job is once they are activated, they release antibodies. By the way, there is a small percentage of them in the blood. Mostly you find them in the lymph. So B cell receptors, see they have a constant region, they look like a Y, and they will have two antigen binding sites. Again, another image of it. T lymphocytes, you have helper T cells or cytotoxic T cells. Cytotoxic T cells have the TCRs. Next to each TCR, you have CD8. Helper T cells, we actually can have type 1 and type 2, but in this class, I'm going to just say helper T cell recognizes and decides. Type 1, if they get activated, they will activate cytotoxic T cells. If type 2 gets activated, they activate B cells. If both of them get activated, then your system produces both a cytotoxic or cell-mediated response and antibody-mediated response. But to make it simple, in this class we're going to just say helper T cell gets activated. This is how it will activate a B cell. This is how it would activate a T cell, cytotoxic T cell. We're going to keep it simple. Ignore the little nuances. And as far as the receptors, see, this is the B cell receptor, how it has a constant region that goes into a fork and has 2 antigen binding sites. All of the TCR or T cell receptors, regardless of whether it's on the helper T cell or cytotoxic, will have 1 antigen binding site. What makes them different, obviously other than what the mechanism is inside the cell, is next to each of these, they will either have a CD4, and that makes it a helper T cell, or a CD8, and that makes it a cytotoxic T cell. Now, since these are randomly made, so in the antigen binding formation or synthesis department, that artist has been told these are your tools, don't question it, just keep making variation. Just anytime you make something new, mark it in the book, don't ever go back to that recipe. So don't repeat it, we're just gonna make 1 set of 3 musketeers for each of those specific antigen binding sites. When you give freedom to the body to create any antigen binding site that they have, what is a possible danger of that? What do you have on the surface of your cells that is unique to you? Your own antigens. Or you have other molecules on the surface of the cell that look like an antigen. So that department has been given freedom. Don't question it, just make it. We'll take care of it. So B cells, cytotoxic T cells, helper T cells, before they are set out into the world of your blood, humor, and all of that, they have to go through a checkpoint. And that checkpoint, we refer to it as a clonal deletion process. Why clonal? Because remember, when they get activated, they create clones of themselves. So what you are doing in this process, you're checking to see whether this one should be sent out there. Before they're fully sent out there to say you can do whatever you want to do, is they are brought in front of every possible marker and antigen that can be found on the surface of your cells that they might react to. Do you recognize this? No. This? No. This? No. This? No. Fantastic. You are ready to proceed out. If they recognize, then immediately they're shut down and they go through a process called apoptosis. So that's the word over there. Apoptosis means cell death program. So it initiates that cell to kill itself. So that B cell, this is the example of B cell. That B cell that recognized something on the surface of your cells as, uh-oh, it's gonna die. And when that dies, all of the possible clones of it will die also, because that will never be made again in your lifetime. B cells do this in bone marrow before they are released and they're fully functional. Cytotoxic T cell and helper T cells go to your thymus. That's why we call them T cells. And they do this clonal deletion process over there too. A person who their body doesn't do this properly, they can have an autoimmune disorder. And sometimes later on in life, something happens that this process gets finicky, or some of your cells that shouldn't recognize an antigen on your body, for whatever reason, even that partial match, they consider it as a full match and they get activated. These are when the person has lupus, Crohn's disease, things like that. So something has been made that causes their own immune system to attack their own cells. Okay? we're gonna look at, I go through that here today. I'm gonna leave it here. All right. I know, brain is like full. So this is, I know, I spent a lot of time on this, but honestly, this is the key thing for being specific. So next time on Monday, we'll go through what happens to the helper T cell, what happens to B cell, cytotoxic T cell, then we kind of see what is the activation of each of them is bringing on. We'll finish with classical complement system, which is after B cells are activated. So we'll do that on Monday. Yeah, I'll let you guys know. And then on Wednesday, we'll finish the immunity and vaccines and all of that. So we'll have something. Might be a little, but I might not have enough time to review on Wednesday, but I think your brains cannot accept any more information right now. If you can, let me know. I'll go talk about macrophages. Okay.
You gave us an hour already.
All right. Okay, guys.
That's the restroom.
Hey, to be or not to be?
It's always B.
Yes.
Hopefully. You choose that one, then you will be.
Yeah, it's like you can go
What's that?
so far.
It's like the hallway.